Hi, okay, so thanks for joining us today. So today we're going to be talking about how to explore infectious disease data in our resource ClinEpiDB.org. So my name is Sheena Tomko, and I'm joined today by Danica Helb and Brianna Lindsay, who's our senior project coordinator, and we're really excited to talk to you about our resource. So just to get started, in case you're not familiar with GoToWebinar, the way it works is that all participants are automatically muted just to make sure there's no accidental interruptions during the presentation. But we are happy to have you ask questions throughout the presentation. And the way that you can do this is to look for this questions panel in your GoToWebinar control panel. And your control panel may look a little bit different than mine, depending on what your computer operating system looks like, but there should be a drop-down menu for questions, and you can type your questions here and we'll receive them. So the way this is going to work is that as you're typing your questions, we'll try and answer them as we go along during the presentation. So just before we really dive into it, I want to go ahead and get and do our acknowledgements. So ClinEpiDB, we're part of a larger group called ViewPathDB, which hosts data on eukaryotic pathogen, vector, and hosts. And our group is funded by a variety of different groups, but the Bill and Melinda Gates Foundation is the main funder of ClinEpiDB. And this group you see over here to the right is our larger group, and we're really excited to share our resource with you today. So first, what we're going to be talking about today is what exactly is ClinEpiDB.org, and as part of that, what data do we currently have available for people to access, and then what data are we also expecting to release over the next year. And then for the second half of today, it'll actually be a live demonstration on our website, and we're going to be covering the different features and tools we have to really help you explore the data. And so what that includes are things like the study page to help provide context to the data, the search wizard and analysis apps which help you visualize the data on our website, how you can download data from our site, and finally save strategies so that you can share your findings with other people through our website. So first, what is ClinEpiDB? Our resource, ClinEpiDB stands for Clinical and Epidemiology Resources, and our goal is to make epidemiology data, particularly that around infectious diseases, more fair. And so for those of you who aren't familiar with the term FAIR, it stands for findable, accessible, interoperable, and reusable. And our website does this in a number of ways. So to make data more findable, we offer unique study identifiers. So every study that we load has its own identifier, and you can think of this as similar to a DOI that's associated with a publication. So if you have that unique identifier, you can always find that data on our site. Accessible, so this is what we'll spend the bulk of our time in the second half of the presentation today. So should be showing you all of these features on our live site on how to use the search wizard to look at the data that's there. So here are some screenshots of how data appear in our search wizard, and we show these kind of images for every single variable within a study. The analysis apps that we have for further visualizing data. So here's an example of one of those. And you can see it adds a little bit extra functionality over our search wizard by allowing you to stratify based on other variables. So here we've got one variable that we're looking at on the y-axis, and we're stratifying by different countries with each of these different plots. And then, of course, we offer downloads of the data. For interoperable, I'll be talking about this a little bit more in a couple of slides, but we harmonize the data that's loaded into our sites using ontologies, and that makes data more interoperable and easier to move between studies. And for reusable, our study pages provide a lot of context and methodology for the study, which is of course really important to making sure that you understand a study when you're doing any kind of analysis. And so ClinEpiDB hosts a variety of different study types. So we have a range of observational study designs that are currently integrated into our site, and also some randomized controlled trials. And within those different study types, we offer a range of different data types. And this is a non-exhaustive list. So we have, depending on the study, information on participants' socioeconomic status and household environment, demographics, clinical observations on these participants, sample assays, and these can be things like microbiology testing or hemoglobin levels, things like that. Anthropometry data, which is obviously really important when studying child growth and stunting and wasting, diet and breastfeeding, travel history, and more. And so this list keeps growing with every new study that we integrate as they bring in new types of data to our resource. And what makes it possible for us to integrate all these different types of data is the fact that we rely on ontologies and that we have ontologies underlying our system. So what this means for potential data providers is they give us their data and a data dictionary. So here we have, for example, the GEMS data, the MALI-D data, and the different malaria isomer data sets. And so we get the data and data dictionaries from the providers, and then our team works to do the ontology mapping and essentially create standardized data dictionaries that are really standardized across different studies. So one great example is the variable sex, which is collected in most studies. And so whenever we see that variable sex, we always map it to the same ontology term so that it's standard across the different studies. These standardized data dictionaries then get loaded into our ClinEpiDB database, and they form a semantic framework. And so the data dictionaries our standardized data dictionaries along with the data itself are loaded into ClinEpiDB, and this really underpins what appears on the website and how you're able to interact with the data there. So if you're curious and want to learn more about how we process data as we're loading it, or if you want a written version of what we're going to show you tonight, some of the different features and tools our sites offer, feel free to check out our publication, which is in Gates Open Research. And following this webinar today, you should receive an email at around this time tomorrow that includes a link to our manuscript as well as to our YouTube channel in case you want to send someone else the link to the recording of today's webinar. And so with that, I'll turn it over to Danica to talk about what data is currently available.
Okay, thanks, Sheena. Next slide. So in this slide, we're basically showing the history of ClinEpiDB over time. So ClinEpiDB started around 2 years ago. In February of 2018, we released our first data set, which was the PRISM isomer project, and that's a malaria project. And since then, we have integrated and released a number of other studies. So in December of 2018, we released the GEMS enteric disease project. In March of 2019, we released both the MALI-D enteric disease study and another one of the malaria isomer projects in India. In June 2019, we released Amazonia and South Asia isomers, and we've been continuing to integrate and release a number of projects since that time. Next slide. And so here we're showing a map that is indicating where all of our data points have come from. And so you can see that we've got a number of studies and data collection happening all over the world. And the other thing I want to point out about this map is this is a demonstration of a future feature that we will be integrating into ClinEpiDB so that you'll be able to easily see where studies have been collected and the sample sizes for those studies. Next slide. We have a lot of data currently available on our ClinEpiDB page. So just to take you through some of the data that we have released already and that you can go and explore on your own right now, we have a number of enteric disease studies. So we have the GEMS Global Enteric Multicenter Study and also the MALI-D longitudinal cohort study. Both of these are very large-scale studies that happened in a number of sites around the world. And we also have a bunch of malaria studies. Many of these are from the International Centers for Excellence in Malaria Research isomer projects. And we have also schistosomiasis studies that are coming out of the Schistosomiasis Consortium for Operational Research and Evaluation. Next slide. And we have integrated a lot of data. So here you can see that for our observation level data, we have almost 5 million unique observations. And we have around 700,000 participants that have been, data has been integrated into the study, and also nearly 300,000 household level data points. Next slide. And as I said, we are constantly integrating additional data sets into ClinEpiDB. So these are some of the upcoming data sets that you could look forward to seeing coming out later this year. So we continue to integrate enteric disease studies. And so we're currently working on PROVIDE, which is looking at rotavirus and oral polio vaccinations. WASH benefits, which is looking at water, sanitation, hygiene, and nutrition in Bangladesh and Kenya, and the VITA study looking at the impact of vaccination on diarrhea. We're continuing to integrate additional studies from SCORE on schistosomiasis and a number of additional isomer malaria projects, as well as non-isomer malaria projects. And finally, respiratory diseases, including the perch pneumonia study. Next slide. And so now I am going to do a demonstration on our live site. So just give me a moment to get that set up. Okay, so I am assuming you can see my screen, and Sheena, please let me know if there's any issues. So on this first screen, this is not the one I wanted to show first. So this is what our current live site looks like. So if you go to ClinEpiDB right now, you'll see this homepage, but I'm actually going to be doing our demonstration today on the latest version of our site, which is not currently released, but will be available next week. We're having a release very soon. So this is the site that will be available very soon. And so what you can see when you come to the ClinEpi homepage, the key on this homepage are these various study cards. And so if you look at these cards, each of these cards represents one of the studies that is available for searching on ClinEpiDB. And you can just mouse over and scroll and see that we have a large number of studies that have been integrated and are available for you to look at. Once you get to this section, you'll see some cards are grayed out. And what this means is that these cards are studies that we at ClinEpiDB are actively working on integrating, and these will be available sometime in the near future, but they're not currently available for you to explore. And so some other features that I want to point out is that we've made it easy for you to sort for studies based on their categories. So if you click here, you can see that you can search for different categories of disease, so enteric, malaria, respiratory, or schisto disease studies. And I'm going to click on this schisto to show you how that sorts these study cards. And so now every card that you're seeing is dealing with schistosomiasis. And the other thing that you can do here is you can navigate with this Find Studies. So if you know of the name of a study that you're interested, you can type that in, or you can type in a country of interest or keywords that will sort the studies that way. So for example, if I'm interested in particular in the WASH benefits studies, I can just type in WASH, and you can see that we have that study card here, and it's not available for searching just yet. Similarly, if I'm interested in all studies that collected samples from Kenya, I can type in Kenya here, and all of the studies that come up now are ones where data collection was done in Kenya. So that's an easy way to find data that you're interested in. Ooh, I'm not sure what happened there. Sorry. Sorry about that. I got kicked out somehow. Okay. Okay, so if you move down further down the home page, we can also see all the way to the right, there's this news panel. And so this is a great way to keep your eye on when we're having releases, what data sets are being released, and also new features and functionalities to the website that we've developed. And then scrolling down the home page, you can get some example searches. And so if you're looking for ways to navigate through data and you just want some pointers, you can sort of click on one of these and follow along and see how we here at ClinEpi have handled a particular search. And then we also have some examples of our visualization tools. And so Sheena will be doing a live demo of how to use the visualization tools. But again, if you want an example of how to use these, you can click here on the home page. Some other key things to talk about from the home page are this About button. So here you can click on FAQ and come to a list of frequently asked questions. And this is a great place to get information that is asked of us quite often, such as, can I do my own secondary analyses? And if I do that, how do I cite the data? How do I go ahead and submit data for inclusion on the ClinEpi site? Things like that. We also have this tutorials and resources section. And so we have a lot of internal resources. So tutorials that really walk you through how to get started with ClinEpiDB. And we also have a few video tutorials here. We also have a link here to all of our workshop exercises. So we do a lot of outreach activities and run multi-day workshops, and you can access all of the exercises right here so that you could work through on your own and really learn the same things that we're teaching in our workshops. And finally, we have a compilation of external resources so that you can see how to really teach yourself data science using Coursera, things like that, different websites that do data visualization and modeling. So just some really cool sites that we'd like to share with the global health community. And then you can always click this ClinEpi button right here to bring you back to the home page. And so one more thing that I want to show you from here is that you can do bulk downloads of the data for analysis on your own computer. So if you're good at using R or SAS or other programming languages, you can just download the data here and perform that analysis on your own. So one thing is that there are often data access restrictions in place, and so you will, if you click download the data, you'll be asked to log in and submit a data access request. And logging in is really easy, just quickly setting up a user account and clicking this button will take you right through that process there. So the download data button on the home page, that's to download all of the data from that particular study in bulk. So the other key thing when thinking about EPI studies is that the context of the study really is important. So you can't just look at the data without understanding the way that that data was collected and the reasons why they the study team collected the data in that way. You really do need to understand the background for the study. And so we've compiled that all onto the study details pages. So if you click on the header of one of these cards, that will bring you to the study details for that particular study. And so here we can see for the GEMS 1 study, it's a case control study design, and we have compiled a list of key publications. So we really recommend that you get started by reading through all of the studies that have already been published and understand what's already been done with this data set. And you can also see the key contact groups and a description of the study, its goals and objectives, methodologies. Here we can see the study sites. And then right here, this is a really important section where we're providing links to all of the original case report forms that were used in data collection for this GEMS study here. And so we really recommend that you access all of these to really understand what each variable means and the context of its collection. And again, principal investigators, so if you are interested in a scientific collaboration, you know who was key in this study, and you can reach out to them and try to set up a collaboration with them. And then I want to show you a study that has a number of data access requests. So the MALI-D study is a very large study that we've had a lot of interest in. And so every time someone requests access to the data, there's a number of things that you can do without having been given full access to the data, such as doing these searches within our site. But if you want to do anything where you're downloading the data, you need to request access to the data. And each time we have an individual who requests access to the data, we keep a record of that. This is I'm still getting used to the new site. I'll show you here. So we keep a record of every data access request, and those also can all be found on the study page. And so it basically gives the name of the requester and their organization and the purpose for which they're wanting to use that data set. And so you can see this, so it's a nice way for the data providers to see the impact that their study is having and the interest that it's generating in the scientific community. And it's just a way to maintain accountability and transparency with who's using the data. Okay, so I'm going to navigate back to the home page. And so now that you understand how to look at the background methodologies for each of the studies, I'm going to start showing you how to navigate through the data on the website. So for my example, I'm going to be demonstrating the GEMS 1 case control study. And what you can see at the bottom of these cards is explore the data. And so for GEMS, we're able to explore the data by participants. So I'm going to go ahead and click that. And this is going to bring up our page where we're able to explore the data. And so I just want to point out at the top, this right here is what we call the search wizard. And so it's basically a way that we've organized the data to make it more intuitive for our users to be able to locate particular variable types. And the other key thing that it does is it's demonstrating the number of participants that meet our filter criteria. So since we haven't applied any filters yet to this data set, we are seeing that we have about 22,000 participants. And so that is a number of participants that were in the GEMS 1 case control study. So in this first box, we can select whether a participant is a case or a control participant. And so something that I want to point out now is that this variable would not be important for a longitudinal study, for example. So depending on the study design, the exact boxes that are going to show up in the search wizard might be different than what I'm showing you right here. So if you navigate to geographic region, we can now see that we have data on the different study sites. And so let's say I'm only interested in subjects from Bangladesh. I can go ahead and click that there. And so what you're seeing now is that we have 3,859 participants. And so that means that of the 22,000 total GEMS participants, about 3,800 were coming from Bangladesh. And you can see down here too, if I look at this data here, we can see that that is in fact correct. There are 3,859 participants from Bangladesh. And we can then navigate to other categories of data. And so now we're looking at data on the households. And so these types of variables are going to be things like the construction materials of the house, or the source of the drinking water, or socioeconomic factors, the income level for that particular house. So those types of variables. So a couple of things that I want to show you here are we have a number of households. And so what we've done is we've organized them into this hierarchy of different variable classes. And so these are all of the categories of household level variables. And if I click expand all here, that will open up this tree and you can actually see the variables that we have. So for example, we have a variable on the type of floor material that the houses were constructed out of. So if I click that variable, that brings up all of the different types of floor materials that were found in this study. And so something that I can see here is that I've got a number of these values that are grayed out here. And so what this is indicating is that in the participants that I've selected, and then in this case, that is the participants from Bangladesh, we didn't have anyone who lived in a house with a carpet floor or a palm or bamboo floor. But we did have participants who had cement floors, ceramic tile floors, dung floors, earth floors. So that is one key thing that I can see here. And so we can navigate through here and get a sense of all of the types of variables that were collected. I see some variables on the drinking water source. For example, this one here, water trips daily. If I go ahead and select this one, this is now going to be a continuous variable, not a categorical variable. And so the difference in the display here is that now we're seeing a histogram of this data instead of categories that can be selected. And so for here, something I want to point out is that you see both red bars and gray bars. And so the red bars indicate the data from our selected participants. And again, the selected participants are the 3,800 participants from Bangladesh. And the gray bars are the data from all of the participants in the study who are not selected. So participants from all of the other study sites. And I can select data here by dragging. So I can just drag and highlight, or I can use these boxes here to type in exact values if I want to do it more accurately. And so I'm applying a second filter now. And so now we can see that my number of selected participants has dropped from 3,859 to 2,733. And so what this is indicating is that we have 2733 participants from Bangladesh who made between 1 to 5 trips for water each day. And so you can just navigate through and continue to apply more filters in this way. Moving on to participants, the variables that are found here are types of variables and data that don't change over time. For example, the sex of a participant or their birth date. And again, I can expand all to see all the variables, or I can also type in this find a variable box here if I want to find a particular variable. So if I'm interested in the sex of these participants, I can just type sex in that box and it will bring me right to the variable that I'm interested in. And then for observations, these types of data are data that can change over time. So for example, the participant's height or weight will be different each time the participant is measured. We also have data on the lab test results and, for example, the microbiology test results that they could be positive for E. coli on one day and not another. So these are the types of data that can be found under observations. And I'm just going to expand all and just, again, just peruse through this list and get a list of understanding of the type of data that is available in the GEMS study. And so if I've been applying filters and I don't really remember what filters I've applied and what types of subjects I've selected, I can always click this green filter icon right here. And so this will bring up a list of all the filters that have been applied. So I can just easily remind myself that that 2733 indicates participants who met these two criteria, that they were from Bangladesh and made from 1 to 5 water trips a day. And within here, I can also remove filters. So if I'm no longer interested in the number of water trips, I can just click this X and remove that. And now I'm back up to the 3859 Bangladesh participants with no other filters applied. Okay, so now that I've shown you briefly how to navigate through all of this data, I'm going to remove all of the filters. Oops. And I'm going to take you through a question that I'd like to answer here. So in this example, I am interested in determining how many Giardia positive cases in the GEMS study had at least one matched control that was Giardia negative. So I'm going to do a stepwise approach to answer this question for us. So since I am first interested in finding out Giardia cases, so I'm going to navigate to case control and select case. And so now I've got around 9,000 participants in the GEMS study that were cases. And the other thing that I'm interested is that I want these cases to be Giardia positive. So let's say I don't know where to look for data on Giardia. I can just go and type that in this find a variable search box right here, and that'll take me to all of the variables that have Giardia data. And so I know that it's in this one here. And so what this has brought up is a what we're calling a multi-filter. And so basically these are the analytic microbiology test results for different eukaryotic pathogens such as Giardia. And so this allows us to select more than one eukaryotic pathogen at a time. But for this example, I'm only interested in Giardia. So I'm going to click yes. So I'm selecting participants who were positive for Giardia by ELISA. And so applying these two filters together, so we're now finding that we have 1,700 cases who are also Giardia positive. So you can see that we've reduced the number of participants down from 2,200 down to around 9,000 cases, and now to about 1,700 Giardia positive cases. Okay, so now the next step that I am interested in is figuring out how many of these 1,700 Giardia positive cases had at least one matched control that was negative for Giardia. So to find the matched controls, I'm going to use this related case control step in the search wizard. And the first thing I have to do is click to enable the search. And so I can either keep or remove participants based on what I'm selecting down here below. So there's many ways to approach this, but what I'm going to do is I'm going to keep the participants who were Giardia negative. So I'm going to first select keep here. And I'm again going to look for the Giardia data, and it's under this eukaryote multi-filter. And now I'm selecting subjects who are not positive for Giardia. So select no. And what that is now showing is that we have around 1,200 Giardia positive cases who had at least one matched control that was Giardia negative. So I've answered the question of how many cases had at least one match control that was Giardia negative. And I can now click this view participants button. And this will bring us to the results table. And so this is basically giving the results of all of the participants that have been selected in my search. And so you can see that this table is pre-populated with several variables that you may or may not be interested in. But one thing I want to point out here is that for each of these different columns, you can see a histogram. And if you click on this histogram, it will bring up a distribution of that variable amongst your selected participants. So if I here have opened up a histogram on country, and so I can see that most of the Giardia positive cases who had at least one matched control that was Giardia negative were coming from Mali. I can also choose to add additional columns to this table. So if I'm not seeing data that I'm interested in, I can just click this add columns button and then open this up, and this allows me to add a number of different variables. And so I can navigate through here and try to find what I'm interested in. For example, I want to figure out if they've been given untreated water, or if they were given antibiotics before their stool was collected. So I can just go and click to select those variables, and then click down here to update those columns. And now that data is showing up in this as well. So I can click given untreated water, and so we can see the distribution of which participants were given untreated water. Okay, so something else that I want to point out from this results table is there's also this download button here. And this download button differs from the download button on the homepage that I showed you earlier. So what this download button is going to do is download the search specific results. So instead of getting the results from every row of data in the study, we are now only getting the results from the rows that meet our search criteria. So this is really helpful when working with very large data sets. We don't necessarily care about most of the data in that data set, and it's just also better if you're only interested in looking at data from one country. It's just better to not download all of the data, but only download the data that you're actually going to use. So you can go and click that download button, and again, the different studies have different data access restrictions. The GEMS case control study has some restrictions, and so they're requesting that you log in before you're able to download that data. So I'll just demonstrate what this looks like here, logging in. And so now that I've logged in, I can choose if I want to download participant level data or household observation sample level data. I'm going to download that participant level data. And it's going to ask me about what columns I care about. And so same thing, I'm just going to go through this list and figure out what data is interesting to me. So let's say I'm interested in the breastfeeding data, and I am interested in, let's see, I'm also interested in the drinking water sources. So I want to figure out all of these different drinking water sources. So I can just select the variables that I'm interested in. And then once that's done, I can choose the type of file that I would want. So I personally like working with CSV files, so I would click that and then click this Get Participants button, and that will kick off the download. So you can download subsets of the data that you're interested in. Okay, and that's it for my demo. So Sheena is going to now take you through a second demo.
Thanks, Danica. I'm just changing to mine. Okay, you should be able to see my screen. So for this second part of the demo, I'm going to focus on the analysis apps and how you can use them. And before I go into that, really, I just wanted to reiterate that. So right now, I'm actually working on what's going to be the version of the site that we're going to release this week, because it has all the updates, same as what Danica was working on. And I just wanted to reiterate that, with the data that's on our live site, you can always, all of the studies that are currently on our live site, you can always, click any of these icons and start exploring the data the way Danica was just showing. The data access request really just comes into play for some of those studies when you try and download the data. So you can always look through all the variables. It's just downloading, which may or may not have restrictions on it depending on the study. So for this part of the demonstration, I'm going to use data from one of the malaria studies that was conducted by the India isomers. So that's, again, the International Center for Excellence in Malaria Research, which those centers are funded by the NIH. And so I'm going to search here, and I'm going to use their longitudinal cohort data. And so this is a study that was done at 2 different sites in India. At one site was in Chennai and the other was in Rourkela. And the goal of the study, one of the goals of the study, was to look at malaria incidence and prevalence in India and look at associations with bednet prevalence and bednet access. And so that was one of their main goals. But for this demonstration today, I'm going to ask a slightly different question of, is there an association between socioeconomic status and bednet access? And so I'm already very familiar with this data set, and so I already know the variables that we're going to be looking at today, just to save time. So we are most interested in one of the variables that this study collected was a derived variable called poverty line. So it was assessing whether a given household was above or below the poverty line. And so you can see here that, it's a roughly 50-50 split between households that were above and below the poverty line. So we're interested in that variable. I'm not going to select anything just yet. And then the other variable that we're interested in is looking at bed nets. And so you can see here there's a few different variables relating to bed nets, but I know that the one that I want in particular is this personal bed net treatment. So this, again, this is what we call a multi-filter variable. And as Danica explained earlier, it's really, each of these is its own variable. We just display them together because they're related to a common question. And this provides some benefit as well when you're trying to do some preliminary analyses. So in this case, we can see that the India isomer, they collected data on, and they asked participants if they had an untreated bed net, if they had a regular insecticide treated net that had been treated more than 6 months ago, or a regular insecticide treated net that had been treated less than 6 months ago, a long-lasting insecticide treated net or an LLIN, an electric bat, some other type of bed net treatment, or no bed net. And so in this example that we're going to be doing, you can see that there aren't really that many people reporting having a bed net. So 47 people report having a net treated less than 6 months ago, 180 report having a net treated more than 6 months ago, and 16 report having an untreated net. And so the analysis we want to do, we're actually going to combine and compare people who reported having any type of bed net to people who didn't report having a bed net. And so to do this, I can just click yes on all 4 of these. Obviously, there's actually no instance where a person reported having an LLIN, and that's why you see this NAN here. But so now, what we've done is we've gone from 397 participants to 0, and that's not actually what I meant to do. But the reason that it's happening like that is because right now this is set to all, so it's finding observations with all of the options selected below, meaning it's only selecting participants who reported having each of these different types of bed nets. So instead, what I can do is change that all to any. And now you'll see my number updates, and I have a total of 246 participants who reported having at least one of these different types of nets. And so that's the filter I'm going to apply here, and I'm going to go ahead and go to the results page now. And so here again is what Danica was just showing you, and if you wanted to at this point, you could download this filtered data through this link. You could add columns here, look at the distributions, but what we're going to do now is look at this Analyze Results tab. And on this page, we have 3 different analysis apps that you can use. The first is a distributions app, and so this is basically another histogram, but the way that this differs from all the histograms you were seeing on the search wizard part of the site is that you can actually stratify by another variable or even another 2 variables. So you can see in this example plot that it's graphing one variable, but it's actually split it. It's stratified that data by some other variable into 3 separate plots. The next app is a contingency table app, and this is the one that we're going to use today. And then the third one is the data summaries app. And so this one, for longitudinal data, what it allows you to do is to look at a variable over time. And for non-longitudinal data, what it does is it lets you look at 2 variables compared to each other. So for this example, we're going to be using this contingency table app, and it really only allows you to do a 2x2 contingency table. And even though we're only looking at one app today, you should know that these apps all have a similar look and feel to them, and they all allow you to select your variables in the same way. And so looking at this one should help you know how to navigate the other 2 apps as well. So one thing to point out before we get into this analysis is that these analysis apps are a little bit different than the downloads in that the analysis apps actually act on all of the data, whereas if you go to download the data from that link that Danica and I pointed out, it actually will only download the filtered data. And so because it's acting on all of the data, what we have instead is this ability to essentially when you filter the data, it creates a derived variable called search results. And so in this case, it's search results of the selected participants. And so this essentially, our dependent outcome variable, it's currently by default set to be our selected participants. And so these are the 2 variables we're going to set to begin with. So in a 2x2 contingency table, you generally have an independent or exposure variable, and you're looking at the association between that and dependent or outcome variable. So for our independent exposure variable, we're going to use that poverty line variable that I showed you earlier. So again, this is one way to assess socioeconomic status, and we're going to select our exposure variable to be households below the poverty line. And for our outcome, we're going to actually leave this as is. So we're going to leave the outcome variable to be our search results and our selected participants. So what this really means is that our outcome variable are all participants who reported having at least one net. So then what we can do is, if we choose to, we can choose to stratify stratify these plots by another variable, but we're actually going to leave it without stratifying for now. So over on the left-hand side, you can see there's a few different tabs. And if we look at the contingency tables tab, this will actually give us the counts and the breakdown of the exact numbers for each category. And it's always a good idea to take a look at these tables, especially in a 2x2 contingency table app where, if you have very low counts, then that can significantly skew your results and your findings might not be reliable. The plot grid tab is where it actually shows you a visualization of this. And so here you can see a visualization that's pretty striking. So on the left, we have the selected participants, which again means participants who reported having some type of bednet. And on the right, we have all non-selected participants, or participants who don't report having any type of bednet. And in green are participants who were not below or above the poverty line, and in blue are participants who are below the poverty line. So we can see here that of participants who report having a bednet, only 26% of them are above the poverty line, whereas 74% are below the poverty line. Whereas if you look at people who don't report having a bednet, you see that only 6% of people are below the poverty line, whereas 94% of this group are above the poverty line. And so this is a very striking finding, and you might think, oh, it's kind of an non-intuitive finding that maybe people who are below the poverty line somehow have better access to bed nets than people who are more wealthy. And if you look at this summary statistics table, you can see this is the only app we have that has any type of statistics on it, but it gives you a p-value, odds ratio, and relative risk, and you can see these very striking numbers. But it's important to always take these stats with not to take them necessarily at face value, because as you know, with epidemiology studies, you always want to make sure you're considering potential confounders. And so if we go back and we want to stratify, often default confounders that people think of are things like sex is often a confounder, or age. And in this case, because poverty might be regional or bednet usage might be regional, we might want to stratify by study site in some way. So here we're going to now choose to stratify by a variable, and we're going to choose one of the variables under geographic region, and we're going to go ahead and choose the study site variable. So if we do this and then look back at our plots, we see a very striking change. So now we see that at the study site in Chennai, we actually have no people reporting to have a bednet, and everyone reports having no bednet, and everyone in Chennai also is above the poverty line in this study. Whereas in Rourkela, what we now see is that there's a pretty even split between the proportion of people below the poverty line is similar both in the group that have bed nets and in the group that don't have bed nets. And so that association that we were seeing before wasn't actually a true association. What we were actually looking at was the fact that Chennai, all the people in Chennai have no bed nets and are above the poverty line. And so if you click on this individual plots, it will actually show you the same plots just one at a time, so you can see them a little bit larger if that's what you are interested in. And so if you do find an interesting analysis that's worth sharing, or if you've subsetted the data in some way and you'd like to share that with a colleague or put it in a manuscript, you can do that by clicking the Save button. You will have to log in with a ClinEpiDB account. It takes a minute to create a ClinEpiDB account. But if I do that here, and that if I sign into my current account actually, and then click Save, I can now name this and save this strategy. And what that does is my strategy will then be available under Workspace in my search strategies anytime I come back to the ClinEpiDB site and log in with my account. The other way to get back to this is you can actually click this share icon, and you will then get a URL, and you can copy this URL and you can send it to your colleagues, you can put it in a manuscript, whatever seems appropriate. This is one way for you to share the exact subset of data that you've been working on with other people. And again, as Danica pointed out earlier, on the homepage, if you want some more examples of this, you can scroll down to the Explore Visualization Tools section. Again, remember, this is not our live site, which is why this section is empty. But these cards are available on our live site for you to look at if you have additional questions. And so I guess at this point, that's the end of our formal presentation. So feel free to ask questions in the chat box through the GoToWebinar control panel. And again, if you have questions at any time when using our site, you can always use the Contact Us link to send us an email. And that's our team, the team who's on here today, Brianna, Danica, and myself, are the ones who get those emails and who respond to them. So I'm not seeing any questions just yet. We had some questions earlier so we did the same presentation earlier today, and some of the questions that we had then included, what does a data request access form look like? So I can show that for a second if people are thinking about other questions that they may have. So here, if I come to a data set where I can view the data, but I can't download it. So say I wanted to look at this GEMS HUAS survey data. I can look through the data. I picked let me go back actually, sorry. So here, if I wanted to say access this and I clicked download data and I needed to submit a data access request, it will ask me to log in. Here I'm logging in with an account that doesn't have access. And this is what the data access request form looks like. So you will be asked to fill out the purpose, whether you've contacted the study team and have discussed this with them, your actual hypothesis and research questions, your analysis plan, and your planned dissemination of results.
Sheena, one question we had earlier today was if participants or if people could use the site for students.
Mm-hmm.
And so I was just going to speak to that for a second. So the question I think from our group earlier today was whether or not the data needed to be approved by an IRB for use in a course, for example. If a student is taking an epidemiology course and needs a data set to practice analysis on, or do some just learning how to use statistical analysis software, we do recommend that you would use a data set that is freely available for download. So a number of the data sets are actually do not require any sort of access request in order to download the data. So those would probably be the easiest for a student to use. However, if they are interested in a specific data set that does have data access restrictions, we do ask that when they submit their data access form, they just indicate that they're a student and they're working on a project, and then that will help facilitate the approval from the data providers. Also, if they're planning on using it for something like a practicum or something where they are planning on actually publishing on the data, we do recommend that each person consults with their individual IRB. In many cases, reanalysis of the data will be exempt, but it is important to actually consult with your own institutional IRB whenever you're planning on doing any sort of human subjects research. So that's our recommendation for just reusing the data, particularly if you're going to be publishing. The other thing I did want to mention was I don't know if we highlighted this earlier, but we do have a data access and use policy that we do recommend that all people who are visiting the site take a look at and read, because it is actually something that you are agreeing to by using the ClinEpiDB site, and it gives you an overview of the types of data access restrictions we have on the data, as well as what constitutes kind of fair use of the data. I think that's it for me, Sheena. I don't have any questions on this side either.
Okay, thanks, Brianna. That was great. And so, yeah, we're at the top of the hour anyway, so I think we'll go ahead and wrap this up. But we appreciate you attending our webinar today, and we hope you have a great rest of your day. So thanks, everyone. Bye.